All right, so you've probably heard about tissue culture before. Maybe in a science class or from those cool videos where people clone plants in jars. But how does it actually work? Let me break it down, step by step, so you can see just how cool and surprisingly simple it can be. First up, let's talk about what tissue culture even is. Basically, it's a method scientists use to grow new plants from just a tiny piece of another plant. Instead of planting a seed, you take a small bit of tissue, leaf stem root, and coax it to grow into a whole new plant under controlled conditions. Step 1. You need to pick a healthy plant. This is your mother plant. Using sterilized scissors scalpel, cut a small piece of tissue, usually called an explant. Think of it as snipping a tiny bit of a leaf stem. Step 2. Sterilization. This is super important. You don't want bacteria or fungi crashing the party. So, you dip your explant in a mild bleach solution, then rinse it with sterile water. Gloves and clean tools are a must here. Step 3. Now, you place the clean explant into a special growth medium. This is like superfood jelly for plants. Nutrients, sugars, hormones. You usually do this in a sterile container, test tube jar. Step 4. The magic happens in a growth chamber under some strong lights at just the right temperature. Over the next few weeks, the tissue starts to grow. First, it forms a callus, a kind of lump of cells. Then, with the right tweaks to the growth medium, shoots start to develop. Step 5. Once you see little plantlets with leaves, roots, you carefully move them to soil, another growing medium. At this point, they're still pretty fragile, so you have to harden them off. Basically, help them adjust to the real world by slowly exposing them to normal air and light. And that's it. You've just cloned a plant using tissue culture. This method is used for everything from saving rare species, producing millions of identical crops, creating virus-free plants for farmers. It's science, but it's also a little bit like magic. So next time you see a row of perfect strawberries, a rare orchid, remember? There's a good chance tissue culture made it possible. Thanks for hanging out and geeking out with me about plant science.